Welcome friends to a new session on genetics. In this session, we will discuss extensions to Mendelian inheritance, gene interactions and other modified dihybrid ratios. Let us see how gene interactions can influence a particular characteristic. According to Mendel's experiments, a pair of genes controls each character, but later discoveries proved that the interaction of more than one pair of genes controls a single character. This is known as interaction of genes or factor hypothesis. The interaction of genes was proposed by William Bateson and Reginald Crundell-Punnett. The key diagnostic is that these interactions produce modified dihybrid ratios. There are various types of interactions and these interactions leads to a different range of phenotypes. Inhibitory factor is another important intergenic or non-allelic gene interaction, but in pleiotropism a single gene contributes to multiple phenotypic traits. Now let us understand about inhibitory factor. As we know, the interactions of genes may involve two or more pairs of genes. In symbol, we could say that expression of one gene depends on the expression of another gene in gene interactions. These interactions produce modified dihybrid ratios. Inhibitory factors involves two pairs of non-allelic genes and it affects the same character. In this, one of the dominant genes suppresses or inhibits the expression of other dominant gene and this inhibiting gene has no visible effect at all. Example, leaf color in paddy. The occurrence and distribution of anthocyanin pigmentation in the leaves of the rice plant is very variable and a very striking feature of the crop. The genetics of this character has attracted wide interest and attention from early time onwards and its mode of inheritance in the individual plant part has been extensively studied. Many geneticists reported the number, role and interrelationship of the genes responsible for the expression of anthocyanin pigmentation in the leaf, which may entirely non-pigmented or fully pigmented. In the later case, the leaf blade may be purple, purple wash or faint purple wash in color. Purple leaf blade has been shown by the interaction of a basic and two complementary genes and all the three genes are essential for the production of purple color. The phenomena is known as autumn leaf color. It is a phenomenon that normally affects the green leaves of many deciduous trees and shrubs take on various shades of red, purple, yellow, black, pink, orange, blue, magenta and brown during a few weeks in the autumn season. The phenomenon is commonly called autumn colors or autumn foliage in British English and fall colors, fall foliage or simply foliage in American English. The purples, reds and their blended combinations that enrich autumn leaf color come from anthocyanins in the cells. These pigments are actively produced at the end of summer and not present in the leaves throughout the growing season like carotenoids. The presence of anthocyanin color in rice is determined by the complementary action of two genes capital C and capital A. The gene C is the basic gene for chromogen production and gene A exerts its activation effect on gene C and convert the chromogen into anthocyanin. The gene C and A comprises multiple allelic series of genes. 3 alleles have been found at A locus and 5 at C locus. They are arranged in the order of dominance as shown in the screen. The expression of anthocyanin color is attributed to the complementary effect of the genes C and A. For distinct coloration in paddy, the presence of C and A and another gene P exists. This is concerned with the distribution of chromogenic substance. The majority of varieties, as a matter of fact, however, possess the gene P in common. As a result, a combination of any of the alleles of C and A loci, it follows the principal color types on the leaf. The expression and occurrence of color in the leaves are closely related to the constitution of genes. The occurrence of coloration in these parts when the genes coexist with any gene combination at A and C loci. When exposed to direct sunlight, the PI gene distribute the color over the entire surface of leaf sheath, leaf blade, collar, ligule, auricle, internode, node and pericarp also. The PN gene distributes the color in leaf margin, leaf apex and entire surface of node, auricle, collar and ligule. The most striking colored parts with the PN and PI are the node and leaf blade respectively. The expression of PI gene is reduced by the presence of a suppressor gene JPI that inhibits the distribution of colors at the center of leaf blade. Thus, at present it may be more natural to consider that the appearance of the new type may depend on the presence of another alleles at the C and A loci. There are multiple alleles at 2 loci, 6 alleles at the C loci and 4 at the A. They are temporally designated as CBN and AE respectively and may be arranged as shown in the screen. As to the action of CBN and AE, CBN may be less potent than CBR and give no sign of anthocyanin coloration when coexisting with A. However, when CBN coexists with AE, the highest potent allele at the A locus, also AE is estimated to have more activation effect than A faint red color is produced.